Hello everyone. So this is the first lecture for chapter 2, and I will take several lectures. I will record several lectures for chapter 2, and the purpose of chapter 2 is to introduce you to, let's say, molecular processes inside cells. And that means we first need to get a mental picture of what a cell is. So we're going to start with a sort of a static view, focusing on the components of cells. And we're going to take a bacterium as an example, because most of what I'm going to say applies equally well to eukaryotes, but those are just typically more complicated than bacteria. But the molecular organization is rather similar, except for the occurrence of organelles in the case of eukaryotes. And so typically what I will have in mind as bacterium is Escherichia coli. It's a model organism in microbiology, and in fact it has also been a model organism for molecular biology. So most of the molecular organization that we know inside cells derives from studies that have been done in Escherichia coli. This bacterium has first been isolated from the human gut somewhere in the 50s, in the 1950s, and since then it has been distributed all over the world and studied by different research labs. And so what does an E. coli cell look like? Well, it's say 2 to 4 micrometer in length, okay, and 1 micrometer. So let's do that here. 1 micrometer is 10 to the minus 6 meters. So it's 1 divided by 10 to the power 6. So it's 1 divided by a million meter. So it's 1 millionth of a meter, okay? And its, let's say its thickness is also several micrometers. One way to think of E. coli is, which is a fairly good approximation if you look at the microscope pictures, is that it's a cylinder. So here you have the cylindrical part in the middle, and its caps are half spheres, right? So in a way the volume is the sum of the sphere plus the volume of a cylinder. Right, and so all these numbers are estimates, because these sizes depend on the growth conditions. So if E. coli grows fast, it's actually a lot bigger than when it grows slowly. So for instance, the volume of E. coli increases exponentially with its growth rate. So if we think about E. coli, so we have this is the membrane, so the plasma membrane composed out of lipids, but also proteins, so there are proteins in there, and there are lipids in there. So lipids are fat molecules, so they don't like water, so they make vesicles themselves. If you would throw them in water, lipids would automatically make vesicles. There's also an outer membrane, which is more open to the outside environment, and the in-between area also contains proteins. So this is the outer membrane, and this is called the periplasm. Okay, all kinds of proteins we find there. And these proteins that we find in this membrane, they exist for, let's say, nutrient import. So all kinds of different nutrients. So you can think about small molecules like glucose or ammonium or phosphate, etc. But there are also, of course, export systems. So products are made. These could be proteins themselves, but also small molecules like acetate or ethanol E. coli doesn't make a lot of ethanol though, but yeast, say the organism that you find in beer or in wine, makes ethanol, and that's the alcohol in these alcoholic beverages. Okay, so inside the cell we have the coiled DNA. Okay, so this is the folded DNA. So that's the chromosome of E. coli. And it's almost positioned in the middle. Of course, there are proteins associated with this DNA, so for instance transcription factors or RNA polymerase making mRNA. For instance, mRNA is made on the mRNA, you have a ribosome, and that leads to proteins, right? So transcription and translation is coupled in bacteria. So while the mRNA is being made, it can already be translated by the ribosome into a protein, and then amino acids, as you know, are consumed in the process. They're bound to tRNAs, and these amino acids are made by metabolism. Okay. So we have the DNA compartment with all the associated proteins, and there are also proteins outside. So most noticeably the ribosome is typically not found in the chromatin department, in the nucleoid department. It's called chromatin in the case of eukaryotes and nucleoid in the case of prokaryotes, so in the case of bacteria. Okay, so some facts about E. coli. It consists of about 1 million proteins. Protein to protein distances are about 5 nanometers, and the diameter of a protein is about 5 nanometers too. So that means that each protein inside the cell is roughly 1 protein dimension away from the other one, alright? So it's really packed, and this protein packing is typically called a macromolecular crowding. So the environment, the inside environment of E. coli does not resemble at all a dilute solution as we typically study in the lab, right? We do biochemistry in the lab, and we have highly dilute protein environments. But this is not at all the case in E. coli. Okay, so about 1 million proteins. And of course these proteins, they have different tasks. So we have signaling proteins, gene expression, associated proteins, there's proteins associated with translation, transcription is gene expression, metabolism, maybe a few others, right? So these are the types of proteins we find. E. coli has about 4,000 genes, and in one particular condition it requires about 300 to 400 proteins are required to make itself, okay? Because one of the miracles of biology is that cells are capable of making exact copies of themselves. And so that means that from the nutrients that they find in their environment, they can double themselves in size and then split, and then you have two daughters that they can double in size and split, okay? So that phenomenon is called, well, it's associated with cell growth, so the cell gets bigger, and then cell division. And this entire process requires about 300 to 400 proteins. Then it expresses, say, a few dozen signaling proteins, a whole bunch of proteins associated with gene expression. So those RNA polymerase, transcription factors, sigma factors in the case of E. coli, and maybe also proteins associated with with mRNA degradation and translation, well, the ribosome, elongation factors, tRNAs, all kinds of proteins and other molecules are associated with that. And then metabolism, which is about 300 to 400. So in total, let's say about 500 proteins or so, or macromolecules are needed in a given environment. And but E. coli has a lot more genes, and that reflects also that in different environments it needs different proteins, so it needs different genes. And that also means that in some environments certain genes are not being expressed, whereas in others they are. And that means that whenever you change conditions, certain genes need to be regulated in expression, some need to be activated and some need to be repressed, so silenced. They have to be turned off, such that no mRNA is made from them, and mRNA is made from other genes that are required now instead. So that means that the cell itself also needs to be aware of its environment, and this it does by signaling circuits. So some of these proteins outside are actually sensing proteins. They bind toxins or nutrients and then induce a response. And that means that induce a response, that means that certain genes are being activated. Their corresponding mRNAs and ultimately their corresponding proteins are being made such that the cell can carry out a new task. Say grow on glucose rather than on acetate or which other carbon source has grown on before. Okay? Now all these proteins, of course, they catalyze reactions. There's a substrate and a product. This substrate or product could also be the protein itself, because some proteins they phosphorylate other proteins or dephosphorylate other proteins. They can modify proteins. What can also happen is two proteins they form a complex with respect to each other. Enzymes involved in metabolism, they may have, of course, multiple substrates, say two substrates in one product, or two substrates and two products. Okay? And these products are then processed by the next enzyme. Okay? So this means that networks, that macromolecular, the protein and biochemical reaction networks exist inside cells. Okay? So metabolism, metabolic networks, signaling, signal transduction networks, etc. So that means that all these proteins are not there, are just not sitting there, they're doing their thing. So each is carrying a reaction, or it's supposed to bind to the DNA, and then such that RNA polymerase can bind to, and then the mRNA is made, and then the ribosome binds, etc. So it's all a dynamic process. Okay, so the static view in terms of components is limited because it's only a snapshot. And the right way of thinking about cells, that in fact the cell is a process. Okay, so there is no moment in time in which these proteins are say sitting still or not doing anything. Continuously this cell and the molecular processes it consists of, they lead to changes of this cell. So in concentrations of molecules, in location of molecules, in the expression of DNA, of genes, in the uptake of nutrients, etc. Okay? So in fact, the correct view of the cell is this. Is a dynamic view, okay? So we need a dynamic view. Alright, and that's the complicated bit, okay? And this is where, say, systems biology comes in. So let's say the previous information on the board was molecular biology and cell biology, so it's the structural features of cells, what are they composed of, what are their names, what are how are they folded, what do they do, so it's sort of a catalogue. of what the cell consists of. But this dynamic view is different. This dynamic view tells you something about the functioning of proteins. So what they do, so their activities and their interactions and their concentration changes. Their concentration changes with time. Maybe even their locations in the cell. And there is this other feature that essentially all proteins in the cell are indirectly linked to all others. Right? Via a chain of molecular interactions. So in principle, when you change the concentration of one protein, this can propagate through the entire network of the cell, right? And what is this network? So the network is composed of molecular components, so the nodes are molecular components. So all the molecules inside the cell and the interactions are the edges are molecular interactions. Yeah, and this is it's a whole list, so it could be conversions, so one molecule is converted into another. It could be binding. It could be, formation of a complex, but it could also be the dissociation of the complex. Etc. So the dynamic view on the cell is really the emphasis that the cell is composed out of a network of molecular interactions. That is what the cell ultimately is. And part of this network consists on the membrane surface. Okay? And part of the network is also in the periplasm. But the major part is inside the cell, so in the cytosol. And some part of that network is equally on the DNA. And the DNA itself is also a protein taking active part in this network, like the membrane and like the periplasm. So the dynamic view of a cell is really all these processes doing their thing in some sort of coherent way, okay? So it's the coherent functioning of all proteins, of all molecules, I should say, inside, I should say, in the cell. Right? So also in its membrane, in its DNA, and in the periplasm itself. So this coherent functioning, okay, that is really the key element of what we associate with life. Okay? And that is the thing that we would like to understand in biology. And part of that was on the previously on the board, and that is writing down a catalogue of all the molecular components of cells. And figure out what their genes are, etc. And all the structure, all kinds of structural features and static interactional features inside cells. So molecular biology typically leads to the structure of the network, so the identification of the molecular interactions and which proteins are active at a certain time. But what these proteins then do together, so concertedly, leading to what we associate with life, that is really strictly speaking, that has never been really on the agenda of molecular biology, because this focused on making this molecular catalogue of life. So when we think about life, then what are the features that we associate with life? So let's make a little list so that we all understand what the challenge is that system biology is trying to tackle. Okay, so what is, so let's say, let's write down some characteristics of life. And let's say versus inanimate, or let's say dead systems, right? Okay, when something is alive, what properties does it have? So that's the question that we're going to just briefly touch on. So one is, of course, that there is this phenomenon of self-repair. So misfolded proteins, error in DNA, these are being repaired. These misfolded proteins, they're being degraded, or they aggregate and make inert pools. So the cell's trying to get rid of them. And DNA errors are typically being repaired as good as the cell can. And there's also cell fabrication. So one of the magical things is that cells can make copies of themselves, but essentially exact replicas. And however, of course, errors in DNA are being made. That is inevitable. So the cell cannot be perfect, but it tries to make as good as a copy of itself as it can. And there is so the self-repair is also in a way related to its self-maintenance. Okay, so let's say a form of robustness of the cell. And the self-fabrication part is, say, growth of the cell, right? And then the other thing is self-adaptation or self-adjustment. Okay, so whenever the environment changes, this cell needs to maintain itself and ideally also fabricate itself. That requires that it can adjust itself. Okay, so this is adaptation. And the biological literature, robustness is sometimes called homeostasis. So there are a few of these key elements of living systems. So their homeostatic properties and their robustness. So they try to maintain the coherence that is required to sustain growth. And at the same time they have to adapt to the environment, and they have to do this equally well to ultimately to sustain homeostasis and sustain growth. So it's these three factors that are the characteristic elements of life. And dead systems don't have that. Okay? So my telephone cannot self-repair, self-fabricate or self-adapt, even though maybe cell phones nowadays are the most sophisticated machines that we have, or airplanes cannot do that either. But are there processes in nature that resemble it? Well, there are some highly complicated processes that have arisen in some sort of, in a similar way almost. And one example is a tornado. So you probably know these things, they can move, and they can move cars in principle, if they're big, etc. If you think about this, this is sort of it has many properties in common with a living system. It adapts to its environment. For instance, it changes itself. There is a certain robustness to it. So when it meets a house, typically the house doesn't affect it and it moves on. And it can even lead to smaller tornadoes. It can even self-replicate, because one tornado can give rise to another one. So many of these properties are almost resembled by these systems. I'm not saying that they're alive, because there is one piece is missing, and that is that there is some DNA template that is being copied and passed on to ancestors. And in a way, this is sort of the, let's say, the book or the catalog or the description of the self that is the DNA. Right. So there is one magical thing I think that is really unique to biology, is that all these living systems that are making copies of themselves, they do this by passing on a molecule that contains all the information that is required to express all the proteins and to sustain and maintain this system. And this DNA is of course continuously being erroneously copied, leading to genetic variation, and this genetic variation then leads to competition, because there is a finite amount of resources. These could be nutrients or space, so different genotypes compete, and then those that can do these three features the best, they win, and their corresponding DNA molecule becomes more frequent in nature, and that process is called evolution. And the fitness of a cell is then associated with how quickly the frequency of its corresponding DNA copy changes in time. If you're unfit, it goes down the frequency. If you're fit, it goes up. And so in a way, evolution, right, is all about these DNA templates. So it's about DNA molecules in competition. Okay, that's really the essential feature of biological evolution, is that species are being selected. In fact, it's not necessarily the species that are being selected, it is their DNA molecules that are being selected. So that also means that the DNA molecules can be selected in evolution that do not, for instance, have all of these features that we associate with life. Okay, because a virus does not self-fabricate, but its corresponding DNA sequence can increase in frequency. And it's the same with a bacteriophage, which is it's a virus for bacteria. But also plasmids. Plasmids, they can not self-fabricate without the presence of the host as the virus, but still plasmids can change in frequency over evolution. So the essential feature of biological evolution is that it leads to a dynamics of the frequency of DNA molecules. And only those systems that are alive, they have this self-fabricated feature. But it doesn't mean that evolution is only about living systems, it's equally about non-living systems like viruses and plasmids. Okay, so this was the first part. So this is the introduction about cellular composition, and that we have proteins, we have DNAs, all these molecules together, they form a network, and it's organized in space in terms of this rod-shaped cell that I told you about. Okay, let's stop here, and then I'll see you next time.